Thanks so much, Bull. It's great to be here. And I wanted to share with you a bit about where Axovant Gene Therapies is today after a year of really tremendous transformation at the company, building not only a very strong leadership team with decades of gene therapy experience, but also a diversified portfolio, which, as we'll talk about in the course of our fireside chat, includes rare monogenetic conditions that we're addressing through gene therapies for conditions like Tay-Sachs disease, like GM1 gangliosidosis, but also attempts to extend the reach of gene therapies to much larger market addressable conditions that we hope could make a major impact in the field of neurosciences and a major difference for patients in areas like Parkinson's disease. Over the course of the last year, and this may be a point that is not as well known about Axovant, is that we've really focused on developing integrated capabilities, spanning the value chain from clinical operations to clinical development to regulatory affairs, even having in-house commercial insight, drawing from folks who have had decades of experience at places like Spark at Novartis, bringing insights early on into the course of our development programs to incorporate patient reported outcomes into our studies, to begin thinking about what the patient journey is actually like, and hopefully early on to make an impact through the design of our very studies on those outcomes with not only dose escalation programs that try to find the optimal dose, but moving beyond that to really understand what is it that drives disease burden in areas like Parkinson's disease and Tay-Sachs. And so we'll talk more about that as well. But last week, we had a major milestone for the company, which was the announcement of data on 2 of our programs. The first was in Parkinson's disease, where we announced data from the first cohort of patients. And this was 2 patients at the 3-month mark, an early but promising sign that we could achieve a significant effect not only on the UPDRS score, which is a measure of motor function in these patients when they're off levodopa therapy, but also in measures of dyskinesia. And this is really a critical aspect of Parkinson's. How can we simultaneously improve the motor function of these patients who are on very high doses of levodopa therapy in advanced stages of the disease, but also have an impact on what is a terrible consequence of advanced Parkinson's, and that's often levodopa-induced dyskinesia, these involuntary writhing movements that characterize the later phases of the disease. And what we found so promising was, in this study, being able to demonstrate impact on both of those parameters. Again, an early sign in a low number of patients, but encouraging and drives us forward to advance the program to an even higher dose with the second cohort of patients. In Tay-Sachs disease, similarly, we were able to demonstrate, and we announced last week, improving enzyme activity, not a measure simply of gene expression or of protein production, but of actual functional enzyme activity, tripling in the CSF relative to baseline with consistently enhanced levels in the serum as well. Again, a very encouraging early result with a very clean safety profile observed in this first patient dosed, a child with severe infantile Tay-Sachs disease. And so we've taken these 2 findings and are very encouraged to move forward with rapid pace, enrolling additional patients in the GM1 and GM2 programs, but also moving forward with the second cohort at a higher dose in Parkinson's disease. So here you see our pipeline today, which has really been built up over the course of the last 12 months. In June of last year, we in-licensed our first gene therapy from Oxford Biomedica, who are, in our view, one of the world leaders in the development and manufacturing of lentiviral vectors. That's the product Axovant PD for Parkinson's disease that you see here, now completed the first cohort of dosing, going up a half log higher in dose to the second cohort. You also see at the top of the pipeline our programs in GM1 gangliosidosis and GM2 gangliosidosis, in-licensed last December from the University of Massachusetts. Again, true pioneers in the development of gene therapies for rare monogenetic conditions, and we're proud to be working with them as well. We additionally have some non-clinical programs that are currently in development using the Silence and Replace platform. This includes AXO-AAV OPMD for oculopharyngeal muscular dystrophy, where we not only silence a mutated form of an allele, but also replace in an optimized functional form of an allele that's resistant to the shRNA that's introduced in that very same vector. So an exciting set of programs, an exciting year ahead of us, and if last year was really about building the foundational elements, building the sort of fundamental building blocks, if you will, that makes Axovant a gene therapy company, then this year, 2019, is very much about execution and focusing on building additional data readouts and reaching those clinical milestones that we hope ultimately will drive value for patients. And if we do a good job at that, we hope build the value of the company as well.
Thanks, Pavan. I'll start by asking a question about your transformation. A couple investors have said that it was actually one of the most remarkable transformations they've seen in biotech. Already you're producing data in gene therapy. So can you talk about the decision as to why Axovant wanted to focus on gene therapy and just rate your progress so far?
Yeah, thanks. It's been an incredible year of transformation for us, and I think you're right to point out the speed with which we've been able to move. Though I would say that we'll be judged far less on that speed than on the real impact that we have on patients and their families, and that's really been the focus of all of our efforts so far. I believe if we can become a trusted partner for patients in conditions ranging from Parkinson's to Tay-Sachs disease, then ultimately the value for the company will follow, and I think shareholders will be quite happy with the result. This is, again, a very busy year for us, and as I mentioned, the transformation that occurred over the last year has really been in a few categories. One is building that leadership team with decades of gene therapy experience. It's been building the capabilities internally to not only have sufficient manufactured product to take into our trials and to dose patients, but also to develop the right relationships with sites, with clinical investigators, with patient advocacy groups, such that we can move with deliberate speed on all of these programs. And then I think, finally, there's been an element that has all happened in the public domain. And for the last 3 years before I started as CEO of Axovant, I was at Roivant, where I'll mention that I was involved in the creation of 6 different private companies, each moving through a similar pattern of really trying to create value from the combination of talent and pipeline products that we believe could make a real difference for patients. And I see a virtuous cycle there. What's been different about this experience over the last year has really been the way in which that's been very public. Every one of those stages, whether it's hiring Fraser Wright, our Chief Technology Officer, one of the previous co-founders of Spark Therapeutics, whether it's the in-licensing of an asset from really one of the world leaders in lentiviral vector development, Oxford Biomedica, all of that has been in the public domain. And so a question that I've often gotten is, well, why did you choose gene therapies? Axovant had previously been known as a company that was making, let's say, an incremental advance in the field of Alzheimer's disease using a small molecule platform to do that. Over the course of last year, we've made a deliberate effort to finally put that chapter behind us, to finally move the small molecule legacy behind us, and that was really completed last month with the formation of and spin out of Arvell Therapeutics, which took our small molecule team and platform with it. But why gene therapies? What is so unique about that, and why is that the direction that I think is so exciting for Axovant and the industry as a whole? In my mind, there's really 3 reasons for it. The first is that there is strong mechanistic rationale across the board for the kinds of gene therapies that we are tackling, where the understanding of the neurobiology is sufficiently detailed that we know, for example, dopamine deficiency in the striatum of the brain is a known characteristic of Parkinson's disease. That's why levodopa has been a core therapy in Parkinson's disease for over 50 years. We know in the case of Tay-Sachs and GM1 gangliosidosis that there is a specific enzyme that is deficient in these children that leads to the severe sequelae of the disease, the rapidly progressive nature, even fatal nature of these conditions. And gene therapy offers a solution that allows us to replace in a very specific and a very targeted way the very factors that are deficient in these patients. So I think the strong mechanistic rationale that yields a relatively higher probability of success, that's really what's gotten, I think, scientifically the field so excited about the promise and power of gene therapies. The second aspect that I think is also worth mentioning is the relatively more streamlined regulatory pathways that we believe could follow as a result of that strong mechanistic rationale, where if we have, in particular, biomarker evidence of disease modification or even reversal in many of the indications that we're going after, that paired with real clinical outcomes could lead to development pathways that involve not hundreds or thousands of patients in some cases, but perhaps far fewer. And I think the level of openness and adaptability to novel clinical trial designs has really been encouraging from FDA and MHRA and other regulatory bodies that we've had the chance to interact with. And I think the third thing is the transformative potential of gene therapies, the idea that a one-time treatment could offer a durable, sustained, and step change effect in these kids with Tay-Sachs disease or with GM1 gangliosidosis. That's really what's I think driven us with such passion and speed towards pivoting the company towards a gene therapy future.
Great. So could you characterize the data so far in Parkinson's disease for Axovant PD and actually Tay-Sachs as well? You've touched on it a little bit, but a little bit more on why these are promising results so far.
Yeah, absolutely. And I'm a physician and scientist by background, so I want to take a measured approach in how we think about these results in this early data set. These were 2 patients that were tested in the lowest dose cohort of Axovant PD, and we had a data readout at the 3-month mark. So it's still very early days. But what we found encouraging was that we started out with the assumption that our focus should be on safety and tolerability, that based on the in vitro work that had been done, we viewed this early dose, this low dose of Axovant PD as lying somewhere in between in terms of relative potency, the lowest and the mid-dose cohorts of Prosavin, the first generation of the vector construct. So our expectations from an efficacy standpoint were fairly limited, and I think this is what led to, I would say, an upside surprise in terms of the effect on the UPDRS Part 3 off score. Now, because it was so early in the study, and because we only had 2 patients worth of data, we wanted to design the study around the most objective measures of efficacy that we could find in the field of Parkinson's disease. Over a dozen drugs in the field of PD have been approved on the basis of a objective, well-validated score called the UPDRS Part 3 off score. The way it works is you take a patient, admit them to a hospital, over a 12-hour period, wash out all of their oral Parkinson's medications, including levodopa. And you can imagine a patient with advanced Parkinson's at the end of that 12-hour period, how rigid they must be. Then a neurologist in that monitored hospital setting sits down with the patient and administers this scale, which is looking at, for instance, measures of rigidity, of tremor, of bradykinesia in each upper extremity, in each lower extremity, etc. And so what we like about the scale is that there's very low inter-rater variability from one neurologist to another neurologist administering it. And over the course of the last several decades, we have a huge body of accumulated evidence of the impact of current Parkinson's therapies on the UPDRS Part 3 off score. In a way, you can think about it as a purist's view of motor function in these patients. What is the actual underlying motor function of a patient when you avoid the confounding variable of background medical therapy? And that's precisely what we were trying to measure. That's what this study was designed around. It was also, by the way, the primary efficacy measure of the previous Prosavin study that was published in The Lancet. So we carried that measure forward here. And what was most encouraging to us was the fact that not only did we see a significant magnitude of benefit, a 25-point improvement on the UPDRS Part 3 off score, that was far higher than even the highest dose previously tested of Prosavin. But more than that, we saw a real consistency of that effect across multiple UPDRS subscales. One subscale in particular that regulators quite like is the Part 2 subscale, which focuses on activities of daily living, the lived daily experience of a patient. And I'll share an anecdote of one of the patients in our own study who described, before receiving the therapy, a feeling of helplessness in the mornings as he was trying to dress himself. He actually relied on a caregiver for help buttoning his shirt in the mornings. And you think about the amount of fine motor coordination it takes to take a button on one side of your shirt and connect that to a hole on the opposite side of your shirt. After the treatment, really being able to do that independently. And that sort of profound effect is getting back to what I talked about earlier, the transformative potential of these gene therapies. So magnitude of effect mattered to us on the UPDRS Part 3 subscale, but also the consistency of that effect across multiple scales.
Okay, great. Just in the last minute, what's your perspective on a source of hidden value in the company, or what investors are missing most in your discussions with regards to the Axovant story?
Yeah, that's a great question. And I think that this is maybe something that's more appreciated by biopharma companies who have spent a lot of time in the gene therapy space. The set of capabilities is what I would point to. The capabilities that we have very deliberately built out within Axovant over the course of the last year. Not only the talent we brought on the team, but in functions ranging from manufacturing to regulatory affairs to clinical operations, pharmacovigilance, the ability to run truly global studies if required, and that end-to-end set of clinical capabilities is really something that differentiates us quite strongly. And if I were to comment on the overall strategic landscape in gene therapies today, at least from our vantage point, what we're seeing is that the pendulum may be shifting towards product-based companies, where in many cases, potential strategic partners have thought a lot about building out through acquisition the platform capabilities that they need, and now thinking about, what sort of specific products and what clinical capabilities do I need to bolt on to really ensure that I'm able to advance discovery stage therapeutics all the way through the clinic, and that's something we've been very focused on at Axovant.
Great, thank you, Pavan.